Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 43. The topic is catalysis in the enzyme's active site. Discussing agenda for the topic will be catalysis in the enzyme's active site. So this will be the focus of our discussion in this lecture. Lecture outcome. After watching and listening this lecture, students will know how the catalysis of the substrate takes place at the active site of the enzyme. Introduction. In most enzymatic reactions, the substrate is held in the active site by so-called weak interactions such as hydrogen bonds and ionic bonds. So these interactions are present between the R groups of the amino acids lining the pocket or the cavity in the active site and the atoms of the substrate. The R groups of a few of the amino acids that make up the active site catalyze the conversion of substrate into product. I hope you know very well about the R groups. And the product departs from the active site. So this enzyme substrate complex is established for a short time, and once the reaction is catalyzed, this association between the enzyme and the substrate is destabilized, and the enzyme is available to catalyze another reaction. The enzyme is then free to take another substrate molecule into its active site. Remember, enzymes are not used in a chemical reaction. This is one of the characteristics of the enzymes, that they can be used again and again unless their 3D structure changes. Then they are unable to catalyze reactions. The entire cycle happens so fast that a single enzyme molecule typically acts on about 1,000 substrate molecules per second, and some enzymes are even faster. So the enzyme substrate complex is established and then the reaction is catalyzed, the substrate is converted into products, and then this association is weakened, destabilized, the products are released, and the active site of the enzyme is once again available to bind to another substrate. So all this activity takes place in a short period of time, and there are enzymes which can catalyze or which can break down a substrate into its product at a speed of 1,000 substrate molecules per second. So this is actually the turnover number. Turnover number of an enzyme is the number of substrate molecules which can be converted into products in a unit time, and turnover number can be from thousands to hundreds of thousands. Therefore, very small amount of enzyme can have a huge metabolic impact by functioning over and over again in catalytic cycles. So even if enzymes are present in low concentration, they are still able to catalyze the reaction efficiently because they are not used, and they are not used or consumed in a reaction, and they can be utilized again and again. Figure 1 shows a catalytic cycle involving 2 substrate and 2 products. We will see that on the next slide. Most metabolic reactions are reversible. Reversible means they can move in either direction, either forward or backward. And an enzyme can catalyze either the forward or the reverse direction depending on which direction has a negative delta G. Negative delta G means a reaction which does not require the input of energy. Delta G is a sign which represents free energy. Free energy is the energy available to perform work. So any reactions which have a delta G value means that those reactions are spontaneous reactions. They can occur all of a sudden without the input of energy. So here we have that figure in which 6 steps are shown that represents the process of catalysis. First, the substrate enter the active site of the enzyme and changes its shape. Remember, the shape of the active site will only change according to the induced fit model. In the lock and key model, the active site does not changes. So these are 2 different models. We discussed that in our last lecture, the induced fit model and the lock and key model. So here they are basically showing the induced fit model here. Once the association between the substrate and the enzyme is established, substrates are held in the active site by weak interactions such as hydrogen bonds and ionic bonds. In the third step, the active site lowers activation energy and speed up the reaction. So activation energy is the minimum amount of energy required to convert reactants into products. So what is the function of enzyme? It lowers the activation energy and it helps the reactants to cross that barrier and convert into products. In the fourth step, substrates are converted into products. You can see here the products have been released from the enzyme, and the active site of the enzyme is once again available to catalyze another reaction and to enter into this cycle again. So in the case of reversible reactions, whether the enzyme will catalyze the forward reaction or the backward reaction, it is determined by the value of negative delta G. If a reaction is non-endothermic, then it will be preferably catalyzed by an enzyme as compared to endothermic reactions. Secondly, the concentration of reactants and products also will affect the forward or the backward reaction, whether reaction will proceed in the forward direction or in the backward direction, because the reversible reactions they tends to achieve a state of dynamic equilibrium, and therefore the concentration of reactants and products also affect the momentum of this reversible reaction. The net effect is always in the direction of equilibrium. So if the concentration of product is higher and that of the reactants is lower, then the reaction will move in the backward direction. If the concentration of the reactants is higher and that of the products is lower, then the reaction will move in the forward direction, and this reaction will tend to achieve the state of dynamic equilibrium. Enzymes use a variety of mechanisms that lower activation energy and speed up a reaction. So how the enzymes carry out their activity, they actually decreases the activation energy. Activation energy is the minimum amount of energy required to convert reactants into products. The reactants are usually in the ground state, they are in the stable state, and therefore they must be destabilized in order to be converted into products, and for this destabilization energy input is required. In reactions involving two or more reactants, the active site provides a template on which the substrate can come together in the proper orientation for a reaction to occur between them. So the substrate, it provides a workbench on which the reactants can combine and they can be converted into products. As the active site of an enzyme clutches the bound substance, the enzyme may stretch the substrate molecules towards their transition state form, stressing and bending critical chemical bonds to be broken during the reaction. So all this magic lies in the active site of the enzyme. In the previous lecture we discussed the active site where the R groups of the amino acids interact with the molecules of the substrate, and this interaction results in the stressing and bending of the bonds of the substrate, and as a result the conversion into products is ensured. I have given here this all important diagram again. It shows the active site and catalytic cycle of an enzyme. First, the substrate molecule fits into the active site of the enzyme. The active site of the enzyme grasps strongly, or it puts a strong hold on the substrate, and as a result the bonds present in the substrate are destabilized, and thereby the active site lowers the activation energy and speed up the reaction. Once the substrate is converted into products, this association is loosened, the products are released, and the active site of the enzyme is again available to form enzyme substrate complex with new substrates. So this cycle is repeated over and over again. Because activation energy, this EA means activation, this A is for activation and E is for energy. because activation energy is proportional to the difficulty or breaking the bonds. Distorting the substrate helps it approach the transition state and thus reduces the amount of free energy that must be absorbed to achieve that state. Now, how much energy will be required to catalyze a reaction? It depends upon the type of substrate, the type of interactions or bonds, their stability, their strength, how much energy will be required to break the bonds in the substrate and thus convert the substrate into the products. So what the enzymes do is that they considerably decreases this activation energy because the interaction of the active site of the enzyme with the substrate, it weakens the bonds in the substrate. Therefore, then lower energy will be required to break them. Lower free energy will be required to break them. The active site may also provide a microenvironment that is more conducive, more favorable to a certain reaction than the solution itself would be without the enzyme. So this is yet another significance of the enzyme, that they not only decreases the activation energy, but they also provides an ideal environment where the reaction can move forward. For example, if the active site has amino acids with acidic R group, it may provide a pocket of low pH in an otherwise neutral cell. The cell is neutral, it has a neutral pH. Now suppose a chemical reaction moves forward with more pace if it occurs in an acidic environment, that in those circumstances the enzymes which has amino acids having acidic R group, then such an active site with acidic R groups, it provides a conducive, a more feasible environment in which such a reaction can be catalyzed. So once again, I want to repeat this point, that for example, a reaction which can move forward or which can take place in an acidic environment but the cell is neutral, then enzymes can also provide that acidic environment to such reactions, because for those reactions the enzymes which are usually there have their active sites surrounded by amino acids with acidic R groups. So this microacidic environment in the active site of the enzymes help these reactions to take place instantly. Here, an acidic amino acid may facilitate hydrogen transfer to the substrate as a key step in catalyzing the reaction. Amino acids in the active site may directly participate in the chemical reaction. Sometimes this process involves brief covalent bonding between the substrate and the side chain of an amino acid of the enzyme. Subsequent steps restore the side chain to their original state because enzymes are not consumed in a chemical reaction, although the side chains do establish association with the substrate, but after loosening or weakening the bonds in the substrate, these side chains are once again restored to their original shape so that this enzyme can be used again, so that the active site is the same after the reaction as it was before. The rate at which a particular amount of enzyme converts substrate to product is partly a function of the initial concentration of the substrate. Now, we are discussing here the turnover number. Turnover number is the number of substrate molecules converted into product per unit time. So, how much product will be formed in a given time? It depends upon the efficiency of the enzyme. What is the turnover of that enzyme? It also depends upon the concentration of the substrate. If the substrate are in high concentration, then few enzymes will not make large amount of products in a short span of time, because no matter how much effort an enzyme puts, it cannot go beyond that turnover number. So the concentration of the substrate also affects the amount of product formed. The more substrate molecules that are available, the more frequently they access the active site of the enzyme molecules. However, there is a limit to how fast the reaction can be pushed by adding more substrate to a fixed concentration of enzymes. If the substrate molecules are in large number and enzymes are in short supply, then still there will be less amount of product formed, because at a given time the active sites of all the enzymes will be blocked and the substrate will be in surplus number. So in such a situation, if more product is intended, then the number of enzymes should be increased. At some point, the concentration of substrate will be high enough that all enzyme molecules will have their active sites engaged or occupied. As soon as the product exits an active site, another substrate molecule enters. At this substrate concentration, the enzyme is said to be saturated. So, if the substrate are in surplus amount, enzymes are in less concentration, then at a given time, all the enzymes will have their active sites occupied with the substrate. So we can say that in such a situation, the enzymes are in a saturated form, they are in an occupied form, they are not free to bind with substrates. When an enzyme's population is saturated, means all the enzymes have their active sites occupied with the substrate. The only way to increase the rate of product formation is to add more enzymes. Cells often increase the rate of a reaction by producing more enzyme molecules. How cells produce more enzyme molecules? From where those enzymes come? Remember, majority of the enzymes are proteins in nature. Proteins are made up of polypeptides. Polypeptide is made up of amino acids, and the instructions for the sequence of the amino acids in a polypeptide is conveyed by the DNA. So it is the DNA which stores information for the synthesis of a particular enzyme. This was the end of the lecture. Thank you.